the pill.
Welcome back to the Deep Dive. We're here to unpack complex science, distill it down so it's clear, insightful, and especially for you lecture ready. Today, it's all about a cornerstone of pharmaceutical sciences, how drugs mess with each other's breakdown in the body, specifically enzyme induction. And look, if you're a PhD student getting ready for that qualifying exam, think of this as your custom shortcut. We're aiming for a really solid foundational understanding. So let's dive in.
Absolutely. And understanding drug metabolism, especially induction, isn't just, academic. It's really crucial for figuring out if drugs are safe and effective in the real world.
Right.
This knowledge helps prevent therapeutic failures, when drugs just don't work like they should. And it even shapes how we design new drugs right from the start.
Okay.
So, our mission today, summarize the key ideas about drug interactions involving induction, break down the big concepts, the pathways, and then really shed light on how scientists actually test for this induction potential. We'll be focusing hard on that superstar enzyme, CYP3A4.
Okay, so let's start big picture. Drug interactions, DDIs. Simply put, right, it's when you take two or more drugs together, and the effect isn't just A plus B.
Exactly. It's different from the sum of their parts.
And what's really important for us today is that these metabolism-dependent DDIs, they're everywhere in clinical practice. They're often the unseen hand, you could say.
It's a good way to put it. And that unseen hand often points straight to enzyme induction. It's a really impactful type of DDI.
Okay.
Think of it like this. One drug, the inducer, is like a factory manager. It tells the body's metabolic machinery, speed up.
Right.
It pushes the worker enzymes to churn out more products, in this case, metabolites. So other drugs get broken down faster.
And the consequence for that second drug, that's the key.
Right. That's the crucial part. You see lower blood levels, a shorter half-life, and critically potential therapeutic failure. The drug might just stop working effectively.
And there's a classic, an infamous example of this, isn't there? The rifampicin and oral contraceptive story.
Oh, yeah. That's a really stark one.
Rifampicin, it's an antibiotic, right? It was found to induce both phase I enzymes, mainly CYP3A4, and phase II enzymes, specifically something called UDP glucuronyl transferase. And the result, a huge, 42% drop in the bioavailability of oral contraceptives like ethinyl estradiol, which led directly to unexpected pregnancies.
It really highlights the serious real-world impact these interactions can have.
Yeah.
It's not theoretical.
No, definitely not. So that rifampicin example makes you ask, okay, what is induction at the molecular level? What's actually happening?
Good question. So when we talk about enzyme induction, we mean increased expression of these drug-metabolizing enzymes. More enzyme protein gets made.
Okay.
And that naturally leads to enhanced drug metabolism. Usually this happens because of enhanced gene transcription.
Right.
Basically, the cell makes more blueprints for that specific enzyme.
Got it. More blueprints, more enzyme.
Exactly. And it's important to distinguish autoinduction, where a drug induces its own breakdown, from the more common DDI type, where one drug induces the metabolism of a second drug taken at the same time.
Right, that's the typical interaction scenario.
Precisely. And this is huge for your exam prep because a single inducer, especially one that hits broad-spectrum enzymes like CYP3A4, can potentially affect hundreds, literally hundreds of other drugs.
Wow.
Which is exactly why assessing this early in drug discovery is just non-negotiable.
Okay, let's zoom in on that most valuable player then. The cytochrome P450 superfamily, the CYPs, and specifically CYP3A4.
The big one.
Yeah. So CYPs are these heme thiolate proteins, super important for drug metabolism, mostly in the liver, but elsewhere too. And CYP3A4, it's the dominant one.
Absolutely dominant. It's the most abundantly expressed CYP in human liver tissue. And estimates are it metabolizes something like 50 to 60% of all known therapeutic drugs.
50 to 60%. That's incredible.
It is. And the kicker is its expression is highly inducible. Lots of drugs and other foreign compounds, xenobiotics, can ramp up its production.
Okay. Are there other important inducible CYPs we should briefly mention?
Yeah, definitely worth noting. Others like CYP1A2, CYP2E1, the CYP2C family members, CYP2B6, they all play roles and are inducible. And interestingly, there's CYP2D6, which metabolizes maybe 25% of drugs, but it's not generally considered inducible.
Okay.
But just to circle back for your exam focus, understanding CYP3A4 induction is absolutely paramount because its substrate range is so wide and it plays such a central role in clearing drugs from the body. Its impact is just massive.
Right. So predicting if a new drug is going to induce CYP3A4, that's a major challenge in drug development, isn't it? How do scientists actually test for this? What are the tools?
It is a huge challenge, and they use sort of a tiered approach with three main types of tools. Each has its pros and cons.
Okay, so what's the first line of investigation historically, anyway?
Historically, it was intact animal models, rats, mice, sometimes dogs or monkeys.
Right. And the advantage there seems obvious. You get the whole living system.
Exactly. You get a complete physiologically relevant system, all the complex cell signaling, the full metabolism machinery, it's all there.
But there's always a but.
There's always a but. They're expensive, they're slow, low throughput, and most importantly for human CYP3A4 induction, they often turned out to be, well, pretty poor predictors.
Poor predictors. Why specifically for CYP3A4? What was going wrong?
This is a really critical point, especially for your exam. The core issue is the molecular switch that controls CYP3A4 induction in humans. It's a receptor called the pregnant X receptor, or PXR.
PXR.
Okay, and here's the problem. PXR is significantly different across species, particularly the part that binds to the inducing drug, the ligand binding domain, or LBD.
So the lock is different in different animals.
Precisely. Think of it like that. The inducing drug is the key, but the PXR lock varies. A key that fits a rat's PXR might not fit a human's PXR at all.
How different are we talking?
Well, human and rhesus monkey PXR LBDs are quite similar, about 95% identical, but compare human PXR to rat or mouse PXR, and it's only about 76-77% identical.
Okay. That's a big difference at the molecular level.
It's a fundamental difference. And it explains why CYP3A induction patterns are so different between humans and rodents, but pretty similar between, say, humans and monkeys.
Interesting. Is that true for other CYPs too, or is this a specific PXR CYP3A4 problem?
It's more pronounced for CYP3A4 because PXR is the main driver. For something like CYP3A induction, animal models are actually more reliable. The underlying mechanism involving the R receptor is much more conserved across species.
Okay, so the PXR difference was the big hurdle for animal models predicting human CYP3A4 induction. But then came a breakthrough, right? The humanized mice.
Exactly, a really clever solution. Scientists basically engineered transgenic mice. They knocked out the mouse's own PXR gene and replaced it with the human PXR gene.
So you get a mouse with a human PXR switch.
Precisely. And these humanized PXR mice, they were a game changer for in vivo studies. They provide a much more accurate intact animal model for predicting human CYP3A4 induction. Their response to PXR ligands really mirrors what happens in humans.
That's fascinating. How do they actually use these mice in studies? What's the protocol look like?
Typically, you treat the mice for about 4 to 5 days. That seems to be enough time to get maximal induction. Oral dosing is usually preferred, mimicking how humans take drugs. And controls are absolutely critical. You need a vehicle control, the drug carrier without the drug, a known human PXR inducer like rifampicin as your positive control.
Right.
And specifically for the humanized mice, you'd want controls like mice lacking human PXR or mice with the normal mouse PXR, just to confirm the effect is truly mediated by the human PXR you put in.
Makes sense. And what do they measure?
They look at a few things. CYP3A mRNA levels using quantitative real-time RT-PCR. That shows if the gene blueprint is being made more.
Okay.
They also measure the actual enzyme activity using probe substrates, things like testosterone or midazolam. Measure the formation of specific metabolites like 6-beta-hydroxytestosterone or 1-hydroxymidazolam. More metabolite means more enzyme activity. And you can measure the CYP3A protein directly using Western blotting, but it's generally more time-consuming than looking at mRNA or activity.
Okay, so humanized mice improve the animal model situation significantly, but you still have the throughput issue. So that leads us to the lab bench, right? The in vitro model.
Exactly, cell-based models.
Yeah.
The huge advantage here is the ability to screen a lot of compounds much earlier in the drug discovery process.
And you said these are generally more predictive for humans than the traditional animal models, specifically for CYP3A4.
Generally, yes, because you're using human cells directly. They're also usually more reproducible and cost-effective than animal studies.
But they have downsides too, I assume.
Of course. They're run under lab conditions, not perfectly physiological. They're simplified systems.
Yeah.
And just putting a drug on cells in a dish isn't quite the same as a human swallowing a pill with all the absorption and distribution involved.
Right. So within these in vitro models, what's considered the best? The gold standard?
The gold standard for CYP3A4 induction in vitro is using primary cultures of human hepatocytes, actual human liver cells.
Okay. Why are they the gold standard?
Because they're an integrated model. They're intact human liver cells, retaining many of their natural functions, at least for a while in culture.
How does that work? How do you get them and test with them?
Well, there are basically three key steps. First, you isolate the hepatocytes from human donor livers, maybe from biopsies or organs not suitable for transplant, usually using an enzyme called collagenase to break down the tissue.
Okay.
Second, you let them recover for a couple of days, then you treat them with your test compounds. Typically for 3 days straight, maybe a few different concentrations like say 2, 10, 20 micromolar. And you always include controlled rifampicin as a positive inducer, maybe DMSO as the negative vehicle control.
And third, measurement.
Measurement. Just like in the animal models, you measure CYP3A4 activity using probe substrates. Testosterone or midazolam are common. You look for those specific metabolites. You can also measure the CYP3A4 protein itself using Western blots or the mRNA levels using RT-PCR or Northern blots.
Okay, that sounds pretty comprehensive. But you mentioned variability before. Is that an issue with human hepatocytes too?
Oh, absolutely. It's a huge issue. Donor variability. Where the liver came from, was it diseased? Was it healthy? That can massively affect the baseline CYP3A4 activity and how strongly it gets induced.
So how do you account for that?
You have to use hepatocytes from multiple donors. The standard recommendation is to use cells from at least 3 individual donor livers for any given experiment. That helps average out the high inter-individual differences and gives you more robust, reliable data.
Okay, 3 donors minimum. Good rule of thumb. How do you interpret the results? When do you flag a compound as a potential inducer?
There are generally accepted cutoffs. A test compound is usually considered a potential inducer if it causes more than a 2-fold increase in activity or expression compared to the vehicle control.
2-fold increase, okay.
Or, another common benchmark is if the induction it causes is more than 40% of the induction caused by your strong positive control, like rifampicin.
Right, 40% of the positive control. Got it. But things can get tricky here, can't they? You mentioned something about metabolic masking.
Ah, yes. This is where it gets really interesting and potentially misleading if you're not careful.
How so?
Some compounds are sneaky. They act as both inducers and mechanism-based inactivators of CYP3A4.
Wait, they turn the enzyme production up and shut the enzyme down at the same time.
Exactly. Think about mifepristone, ritonavir, tamoxifen, troleindamycin. There's known examples. Imagine the factory, CYP3A4, is told to build more assembly lines, induction of mRNA protein. But at the same time, the drug itself is throwing wrenches into the existing machines, causing them to break down or stop working in activation.
So the net effect on activity might be 0 or even negative.
Precisely. The net observed activity might not increase, or it could even go down, even though the gene transcription and protein synthesis are being induced.
Wow, so if you only measured enzyme activity,
you could get a false negative. You conclude the drug isn't an inducer when actually it has the potential to induce. But that effect is masked by simultaneous inactivation.
That's a major pitfall, so how do you avoid that trap?
For compounds like these, measuring mRNA or protein levels directly becomes much more informative. It shows you the underlying induction potential, even if the final activity measurement is confounded.
Okay, that makes perfect sense. Measure upstream of the activity. Are there other in vitro options besides fresh primary hepatocytes?
Yeah, there are alternatives. There's an immortalized human hepatocyte cell line called F2N4, developed by Pfizer, which can be useful. And people also use cryopreserved human hepatocytes, frozen ones.
How do those compare?
Cryopreserved cells are convenient, but sometimes they don't attach as well to the culture plates, and their baseline enzyme activities might be lower compared to fresh cells. So pros and cons to each.
Okay, so beyond these whole cell models, there's another in vitro tool that's even more targeted, right? The PXR reporter gene assay. You mentioned PXR being the key switch.
Yes, this assay zooms right in on that PXR activation step.
How does it work? And does it help with that metabolic masking problem?
It absolutely helps with masking. The basic idea is you take a cell line and you introduce 2 pieces of DNA into it, you co-transfect it.
Right.
1 piece of DNA codes for the human PXR receptor. The other piece has a reporter gene, often luciferase, which makes light hooked up to DNA sequences that PXR normally binds to, called PXR responsive elements.
So it's like installing a PXR-specific alarm system in the cell.
That's a great analogy. The inducer drug is the intruder. It comes in, binds to the human PXR you've added, the sensor. This activates PXR, which then binds to its responsive elements on the second piece of DNA, and that triggers the alarm. The luciferase gene gets turned on, makes light, and you measure the light.
Clever. And the advantages?
It gives you a direct measure of PXR-dependent gene activation. It tells you if your drug is flicking that PXR switch. And because you're measuring this upstream event, the switch being flicked, it sidesteps the metabolic masking issue. You'll see the PXR activation even if the final CYP334 enzyme activity is being simultaneously inhibited downstream.
That seems really useful for those tricky compounds.
It is. And studies generally show a good qualitative correlation, meaning if something lights up the PXR reporter assay, it's likely to be an inducer in primary hepatocytes too. Getting the exact quantitative match is harder, but it's a great screening tool.
Okay, so we've got animal models, especially humanized mice, and various in vitro tools like hepatocytes and reporter assays, but the ultimate test has to be in humans, right? In vivo assessment.
That's the gold standard for clinical relevance, yes.
Yeah.
Direct assessment in humans.
The pros are obvious. You see the real effect in the actual system we care about. But what are the cons? Why don't we just do human studies for everything?
Well, they're expensive, logistically very complex. And there are significant ethical considerations, especially when using healthy volunteers who don't stand to benefit therapeutically from the test drug. There's always some risk involved.
Right. Safety first. So assuming you do proceed with a human study, how do you measure induction?
Several ways. One classic method, developed back in the late 80s, is the erythromycin breath test.
Erythromycin, the antibiotic, how does that work?
Yeah. So erythromycin gets N-dimethylated, a specific chemical reaction by human CYP3A4. The test involves giving a tiny dose of erythromycin that's labeled with radioactive carbon-14 on its N-methyl group.
Radio-labeled, okay.
When CYP3A4 metabolizes it, that labeled carbon gets released and eventually breathed out as labeled carbon dioxide, 14CO2. So you measure the amount of 14CO2 in the person's breath. More 14CO2 means more CYP3A4 activity.
Ingenious. Has it been used much?
Oh yeah. It's been used to show CYP3A4 induction, but lots of things St. John's wort. Certain HIV drugs like efavirenz, steroids like dexamethasone, and of course, rifampicin. The main drawback, obviously, is the use of radioactivity.
Understandable. So, what are the safer alternatives?
Using non-radio-labeled drugs that are known CYP3A4 substrates, often drugs that are clinically important themselves.
Like what?
Metabolism is a really common one. Also, buspirone, phenylpines, statins like simvastatin and lovastatin, even testosterone.
And the midazolam example you mentioned earlier with rifampicin.
Exactly. Treating people with rifampicin significantly speeds up how fast their body clears midazolam, about a 2-fold increase in clearance. That directly translates to lower drug levels, reduced Cmax and AUC, and a shorter half-life for midazolam. It's a clear clinical demonstration of induction.
Okay. Are there even less invasive ways? Maybe without giving any external drug?
Yes, that's kind of the holy grail, measuring endogenous biomarkers. Things your body makes naturally that are metabolized by CYP3A4.
Like what, for example?
Well, one is 4-beta-hydroxycholesterol. It's a cholesterol metabolite made by CYP3A4. Studies found its levels in plasma shot up like 7 to 20-fold in people taking certain anti-epileptic drugs known to be inducers.
Wow, 20-fold.
Yeah. Another one is 6-beta-hydroxycortisol, a metabolite of cortisol. Its excretion in urine increased more than 3-fold after rifampicin treatment.
So measuring these natural metabolites in blood or urine could give you a window into CYP3A4 activity without giving any test drug.
Exactly. It's really promising for non-invasive monitoring in the future. Least hazardous approach.
Okay, so stepping back. We have in vitro data, animal data, human data. How well did they all line up? Is there a good correlation overall?
There's generally a decent qualitative correlation, especially for well-studied inducers, meaning if something looks like an inducer in vitro or in humanized mice, it probably is an inducer in humans.
But quantitatively, predicting how much induction?
That's much fuzzier.
Yeah.
Getting the numbers to match precisely between systems is tough.
Why is that so difficult?
Because so many other factors come into play in vivo that aren't fully captured in vitro. Things like the drug's dose, how it's given, how long it's given for, its own pharmacokinetics, how it gets absorbed, distributed, eliminated, where does it actually concentrate in the body? And then there's that metabolic masking issue, again, potential inactivation happening at the same time. All these things influence how much inducer actually gets to the PXR receptor in the liver cells.
Right, the effective concentration at the target site. That makes sense.
So predicting the exact magnitude of induction in humans based solely on preclinical data remains a significant challenge.
Okay. Now, you mentioned PXR, that key receptor, is the switch for CYP3A4. But does it only control CYP3A4, or is its influence broader?
Excellent question. And this broadens the picture considerably. No, PXR doesn't just regulate CYP3A4.
What else does it do?
It's also known to regulate the expression of some phase 2 conjugating enzymes, like UGT1A1, which adds glucuronide molecules to drugs.
Okay, phase 2 enzymes too.
And importantly, it also upregulates drug transporter proteins. Proteins like P-glycoprotein, PGP.
Pharma Power.